few more people coming in. Okay, I think we will start. Hello, everyone, and welcome to this webinar. It's the ISHR ES webinar series, The Beating Edge, and today's webinar is about cardiac fibrosis. And the chairs for this webinar and organizers are actually the early career and mid-career scientists of the ISHR Council. And it's my pleasure to introduce Dr. Jonathan Lambert, who is from University of Padua, who is an ECI, and Dr. Alice Marino, who is from the UC Louvain and is a mid-career scientist, and myself, Dr. Karoline Rypdal from the University of Oslo, early career scientist. So I can just mention right now that we have this Q&A box that we would like you all, if you have questions for our speakers, just put them in the Q&A box. You can do that at any time during the presentations, but we will address all the questions in the end of the webinar, so after our three speakers. And today we have three excellent speakers. We have Associate Professor Arantxa González Miqueo from the CIMA University of Navarra, and we have Dr. Javier Barallobre-Barreiro from the Imperial College London, and we have Professor Sandrine Horman from the University of Louvain. But before we let our first speaker enter the scene, we will have Jonathan, you will have some information for us.
Hi, everybody. So Jonathan here. I know that everyone that went to the World Congress in Japan had a great time in Nara and is looking forward to our next meeting, which is actually going to be our European Congress. And this is going to be in Birmingham in the UK on June 22nd. And just some key dates to mark in your calendar is that abstract submission and conference registration will be open at the end of 2025. ISHR ECI Travel Awards will be open in January of next year. The abstract submission deadline is going to be March 29th. And this is the conference website. I will put this on the chat so you can find all these further details. And that's really all the news and updates we have for you. And Karoline will go ahead and introduce today's first speaker.
Okay, thank you, Jonathan. So the first speaker of today is Associate Professor Arantxa González Miqueo from the CIMA University of Navarra. She is the director of the program of cardiovascular disease there and a group leader of the Spanish Cardiovascular Research Network. She has been studying mechanisms of fibrosis in heart failure and myocardial remodeling for many years. And in 2023, she was appointed Fellow of the Spanish Royal Academy of Science. So Arantxa, we are very excited to hear your presentation for us today, which is entitled Cardiac Fibrosis in Heart Failure, Clinical Relevance and Non-Invasive Diagnosis. So please, the stage is yours.
Thank you very much, Karoline, for such a nice presentation. Let me just share my screen. Go into presentation mode. Okay. So yeah, thank you very much and good afternoon, everybody. And I would like to start by thanking the chairs of this session for organizing this very interesting webinar focused on cardiac fibrosis and of course for counting me in for this presentation. And first of all, I would like to remind you all that the extracellular matrix plays a key role in maintaining the structure and morphology of the heart and also in keeping cardiomyocytes together, aligned and beating together indeed. In physiological conditions, if we look, for instance, at this myocardial tissue sample from a subject who died of non-cardiovascular disease, we can see that the extracellular matrix is quite small in quantity. As we can see here, collagen stained in red. But if we looked at the molecular level, we would see that indeed this extracellular matrix is composed by a complex network of molecules involving not only the fibrillar collagens, but also other types of collagens, glycoproteins, glycosaminoglycans, glycoproteins, cytokines, and different growth factors. In response to different types of injury and disease in the heart, there is an alteration in the composition of the extracellular matrix of the myocardium and an accumulation of collagen and other molecules that lead to the development of fibrosis. And this fibrosis can have a detrimental impact. On the one hand, it alters the physical properties of the myocardium. Since collagen, it is a very stiff molecule, it increases the stiffness of the left ventricle and also of the atria. It alters these cellular interactions and the tissue architecture that we just mentioned was essential for keeping the structure of the heart. And it also alters the function of the extracellular matrix as a reservoir for different cytokines and growth factors. This can lead to an impairment in left ventricular diastolic function, primarily at this function of the diastolic function. But in very severe conditions, it can also affect and compromise contraction and systolic function. It also facilitates the development of cardiac arrhythmias, and it can also have an impact on oxygen perfusion, contributing to the development of hypoxia and even to ischemia. And overall, it contributes to impaired clinical outcomes in patients with different cardiac conditions. Indeed, there are several studies showing this type of impact of cardiac fibrosis in heart failure. For instance, in this study performed in patients with heart failure with preserved ejection fraction, we can see how histologically assessed collagen volume fraction fibrosis is associated with an increase in the E/E prime ratio, which is an index of diastolic dysfunction. And in this large study performed in patients with cardiac conditions in which fibrosis was evaluated by cardiac magnetic resonance measuring the extracellular volume, we can see that the higher the extracellular volume, the higher is the risk of hospitalization for heart failure and even mortality. If we look into, with a little bit more of detail, into this myocardial fibrosis, excuse me, we can see that it is quite a heterogeneous process that can appear in different forms. We can have different small or large foci of replacement fibrosis in which collagen occupies the area that was left by dead cardiomyocytes. And in these large scars, for instance, are the typical examples of replacement fibrosis after a myocardial infarction. We can also see reactive fibrosis in which there is a thickening of the physiological strands of collagen that usually maintain this structure of the heart and surround individual cardiomyocytes. And finally, we can also find an accumulation of collagen surrounding intramyocardial blood vessels, which is called perivascular fibrosis. And it's also important to take into account that these types of fibrosis are not mutually exclusive, and that indeed we can find a combination of these types of fibrosis within the same patient. Therefore, it is important to take into account that myocardial fibrosis is an heterogeneous and dynamic process that, depending on the triggering factor and on the stage of evolution of the disease, we are going to find different cell types and different molecules involved in the development of this fibrosis. Therefore, the one-size-fits-all strategy is not going to work in the treatment or when we address myocardial fibrosis. And we need to perform a careful phenotyping and evaluation of fibrosis. Indeed, it's also important, I would like to highlight that fibrosis is at least partially reversible and can be prevented and even regressed. And a lot of effort is being made in developing specific antifibrotic therapies, which right now are missing in the cardiovascular landscape. Unfortunately, for the sake of time, I will not be able to get into detail in the aspect of therapeutics of fibrosis. Touching briefly on the process of fibrosis development, in physiological conditions, cardiac fibroblasts are in a quiescent state. but in response to stress and different types of injury, there is an activation and differentiation of fibroblasts into different phenotypes, including active fibroblasts and myofibroblasts. These profibrotic fibroblasts that produce an excess of collagen, in particular in the heart, the most abundant type of collagen is collagen type 1, they release collagen as procollagen type 1, with carboxy and amino-terminal propeptides that need to be excised by specific proteinases to render the mature collagen molecules. However, when we analyze the impact of cardiac fibrosis, not only the quantity of collagen fibers that we have is important, but how those molecules are organized and bound to each other is also important. And this is the last step in the process of collagen fibers maturation, which is the process of collagen cross-linking. This collagen cross-linking is mediated mainly by the enzymes of the family of lysyl oxidase, LOX, and it renders collagen fibers stiffer and more resistant to degradation. And we and others have shown that indeed this increase in myocardial collagen cross-linking in patients with heart failure is associated with an increase in parameters assessing left ventricular stiffness, such as the E to E prime ratio or the pulmonary capillary wedge pressure. A few years ago, indeed, trying to understand a little bit more the impact of this collagen cross-linking, in a small cohort of patients with heart failure of hypertensive etiology, where we had access to myocardial biopsies, we found that approximately 70% of the patients with heart failure presented high myocardial collagen cross-linking, whereas the remaining 30% had similar levels to that of controls. Interestingly, the degree of fibrosis, the total fibrosis, the collagen volume fraction, was similar in both groups of patients. However, the prognosis of the patients was different. Patients with a higher degree of cross-linking, with a higher collagen cross-linking, presented a higher risk of hospitalization for heart failure. Therefore, since I have shown you that cardiac fibrosis has an impact on the prognosis of patients and on the evolution of cardiac dysfunction, it is important to be able to, first of all, carefully characterize fibrosis, diagnosis, and also monitor its evolution in response to therapy. And the first challenge that we face is that cardiac biopsies are not routinely available for the direct assessment of cardiac fibrosis. And therefore, we need to rely on surrogate biomarkers. In this regard, we have multimodality cardiac imaging techniques, and in particular, cardiac magnetic resonance, that can give some information about tissue fibrosis, about cardiac fibrosis, both focal and diffuse fibrosis. And the other approach that we can follow, and is where I'm going to focus for the second part of my presentation, is the use of circulating blood biomarkers, which are usually easy and affordable and simple to apply to large populations for screening. But reality is that this has been a very active area of research, and a high number of biomarkers have been proposed, although very few of them have reached the clinical arena or are being used even in clinical trials. And this is probably due to a proper lack of validation and not fulfilling the ideal characteristics of a biomarker. Because what do we expect from a biomarker of fibrosis? Well, of course, first of all, it should reflect the alteration of interest, in this case, fibrosis, and it should have a clinical relevance for diagnosis, prognosis, or for therapy monitoring. And importantly, from a technical point of view, the method of measurement needs to be robust, easy, affordable, and reproducible. And along the last year, I think that enough evidence has been gathered supporting a potential role for collagen type 1 derived biomarkers for the assessment of cardiac fibrosis in heart failure. So I already mentioned that the first step in the processing of collagen is the maturation from procollagen into collagen. And in this process, the carboxy terminal propeptide, P1CP, needs to be necessarily excised from the molecule of procollagen. And we showed already almost over 25 years ago that this biomarker of fibrosis, P1CP, is associated with histologically assessed fibrosis in endomyocardial biopsies in patients with hypertensive heart disease. And this association was even stronger in patients in a more advanced stage of disease who had already developed heart failure. We have obtained similar results in patients with other cardiac conditions, such as dilated cardiomyopathy and heart failure with reduced ejection fraction, also finding this association with P1CP and histologically assessed fibrosis. Here again, the association was stronger in those patients in a more advanced stage of heart failure. Interestingly, this biomarker can be also useful for stratifying the risk of patients or their response to therapy. In this study that we carried out in collaboration with Professor Vallès-Canis in Barcelona, in over 600 patients with heart failure with reduced ejection fraction, where we evaluated left ventricular reverse remodeling after 1 year of guideline-guided medical therapy, we found that indeed those patients with low P1CP upon recruitment, so those with less fibrosis, were those who experienced a better reverse remodeling in response to therapy. For instance, these were the patients that reduced more their diameter size and volumes, as shown here in the left end diastolic diameter. Also, patients with low P1CP were those who improved more their left ventricular ejection fraction in response to the optimization of therapy. And not only that, but those patients with low P1CP, so low fibrosis, had a better outcome during follow-up. They presented a lower risk of rehospitalization for heart failure or cardiovascular death. Interestingly, the patients who improved their ejection fraction, as expected in response to therapy, presented a better outcome during follow-up. But when we introduced P1CP in the equation, we found that this beneficial effect of the improvement of left ventricular ejection fraction, here the blue line, was mostly found in those patients with low P1CP, whereas those patients who improved their ejection fraction but presented a high fibrosis didn't have such a good outcome. We have obtained also similar results in other cardiac pathologies like dilated cardiomyopathy, where those patients with high fibrosis, assessed here by a combination of cardiac magnetic resonance assessed late gadolinium enhancement and high P1CP levels, were those that presented a higher risk of presenting cardiovascular events during follow-up. Interestingly, this biomarker is also useful to monitor the antifibrotic effects of some therapies that have been shown to have some antifibrotic potential, such as mineralocorticoid receptor antagonists. And there are several clinical trials showing this effect on P1CP. Getting with a little bit more of detail in the results from the HOMAGE clinical trial, in which patients at risk of heart failure were treated with spironolactone, we found that spironolactone induced a reduction in P1CP levels as early as 1 month after the initiation of treatment, and that this effect was maintained until the end of the study. Of course, this potential antifibrotic effect was also accompanied by other cardioprotective effects, such as a decrease in the left atrial volume index and an improvement in NT-proBNP levels. More recently, similar results have been found in the Emperor trials, where it has been shown that empagliflozin is also able to reduce P1CP levels, also pointing to a potential antifibrotic effect. We have talked about collagen quantity, but what about collagen cross-linking that we also said it's very relevant? So in this case, we rely on a marker of collagen degradation. Collagen fibers are degraded by matrix metalloproteinase 1, which render a large telopeptide, which is further degraded by gelatinases, and a carboxy-terminal telopeptide, C1TP. The interesting thing about C1TP is that it is a cross-link peptide. And since increased cross-linking makes the fiber more resistant to degradation, the higher the degree of cross-linking, the lower quantity of C1TP will be released by a given quantity of MMP1. And indeed, we showed that patients with increased myocardial collagen cross-linking assessed in biopsies presented lower levels of serum C1TP to MMP1 ratio, existing also a quantitative association, inverse association between myocardial collagen cross-linking and the serum biomarker. This biomarker also has a prognostic value, and it has been shown, validating the results that we obtained in the small invasive cohort in a series with over 230 patients with heart failure of hypertensive etiology. We found that those patients with a higher degree of collagen cross-linking assessed by the biomarker presented a higher risk of hospitalization for heart failure. And indeed, after adjusting by a number of clinically relevant factors, including NT-proBNP, the risk remained twofold higher in patients with high collagen cross-linking as compared to those with normal collagen cross-linking. Therefore, this combination of biomarkers, P1CP and C1TP, can identify a profile of fibrosis characterized by severe deposition of highly cross-linked fibers, which is associated with especially poor prognosis. In this example, I show you how the patients with this profile of fibrosis, as presented here in the orange line, are at a higher risk of atrial fibrillation recurrence after ablation. And here again, after adjusting for clinically relevant factors, the risk remains threefold higher in patients with severe deposition of highly cross-linked fibers. Interestingly, the biomarker of cross-linking can be also useful to identify those patients who are going to respond better to therapy. If you remember, I just mentioned that in the HOMAGE clinical trial, spironolactone was able to induce a reduction in the left atrial volume index. But when we stratified patients according to our biomarker of cross-linking, we found that in those patients with the highest cross-linking, this effect on the left atrial volume was not found. Indeed, the atrial volume in patients treated with spironolactone at 1 month was a little bit higher than in those treated with placebo. In patients with an intermediate degree of collagen cross-linking, spironolactone reduced the left atrial volume at 1 month, but the effect was lost by the end of the study. And only in patients with low collagen cross-linking, this effect was maintained until the end of the study. And we obtained similar results regarding the impact on NT-proBNP values. Validating these results, we also saw in the ALDO-DHF trial, in which patients with heart failure with preserved ejection fraction were treated with spironolactone. In this study, spironolactone reduced the E to E' ratio, an index of diastolic dysfunction. And when we stratified patients by our biomarker, we found that patients with high collagen cross-linking didn't show this beneficial effect of spironolactone on the E to E' ratio. This effect was already seen in patients with intermediate cross-linking, but it was the most evident in patients with a low degree of collagen cross-linking, supporting an interesting role for this biomarker in the management of patients with heart failure. Although I have focused mostly on collagen type 1, I would like also to remind you that in the myocardium, there are also another type of non-fibrillar collagens that are also very important from a functional point of view. And this is the case, for instance, for collagen type 6, which as you can see in this immunohistochemical staining, is quite highly expressed in the myocardium of patients with cardiac conditions such as hypertensive heart failure. And collagen type 6 releases a bioactive peptide, which is endotrophin. And recent results from the TOPCAT clinical trial have shown that higher endotrophin levels are associated with a higher risk of mortality or hospitalization for heart failure in patients with heart failure with preserved ejection fraction. And interestingly, the predictive value of endotrophin is higher than that of NT-proBNP and of the risk score matching. We validated this finding in different independent cohorts across Europe and the United States, showing this prognostic value of endotrophin 1 for predicting hospitalization for heart failure and mortality, which remained significant after adjusting for the MAGIC risk score. And just to finish my last scientific slide, I would like to point out that with the development in the last years of novel high-throughput proteomic-based techniques for the assessment of serum and plasma, such as proximity extension assays panels or aptamers, this has opened new avenues for studying proteomics in serum plasma, and combining this approach with machine learning algorithms, we are able to gain new insights into the mechanism mediating the action of drugs. For instance, here we were studying the mechanism leading the changes in P1CP induced by spironolactone, leading to the discovery of new molecules that can be playing a role and can be novel pathophysiological mediators in this process. So just to finish, my take-home messages that I hope I was able to transmit are that, first of all, myocardial fibrosis is an heterogeneous and dynamic process contributing to cardiac dysfunction and heart failure development and progression, that it is important to identify, diagnose, and monitor fibrosis through surrogate biomarkers or parameters, and that in this scenario, circulating biomarkers can be useful for this first screening and monitoring of fibrosis. That P1CP, derived from collagen type 1 synthesis, is a suitable biomarker because it's associated with histologically assessed fibrosis. It is modulated by antifibrotic therapies, and it has a prognostic value. And that also assessing collagen cross-linking through surrogate biomarkers, it's important because it has a prognostic value, and it can be useful to identify responders and non-responder to existing antifibrotic treatments. And with that, I would like to thank my team, all our collaborators, and of course, thank you all for your attention. Thank you very much.
Thank you very much, Arantxa. That was a fantastic talk on circulating biomarkers and cardiofibrosis. I know I, for one, learned a lot. So just to remind everybody, we're going to be taking questions and answers at the end of all three talks. So if you want to ask a question, go ahead and put it in the Q&A. But I will go ahead and introduce today's next speaker as he's pulling up the slides. So our next speaker is Dr. Javier Barreiro. He earned his PhD from the University of Coruña in Spain, where he was studying cardiac remodeling in a pig model of cardiac ischemic injury. He then completed a postdoc at King's College in London with Professor Manuel Mayer, where he began studying the cardiovascular extracellular matrix and mechanisms of protein fragmentation in a myriad of cardiac disease models. Currently, Javier is a senior lecturer. He started a lab at King's College, but currently he is a senior lecturer in cardiovascular extracellular matrix biology at the National Heart and Lung Institute at Imperial College. And today he will discuss the beating edge of cardiac fibrosis, focusing on the proteomics landscape of dilated cardiomyopathy. So go ahead and take it away, Javier.
Thank you very much for the introduction. Just checking that you can see my screen now.
We see your lecture notes, so if you could.
You see the notes again.
Yeah.
You can press the swap displays again, I think, Javier.
I don't have it now. That's the issue. So I was looking. Oh, yeah.
Oh,
now I
because
see
we
it.
can see it. Yeah. Perfect.
And I think you see yourself as well now. Do you see it now?
Yep.
All right. Sorry
Perfect.
for the technical issue.
No problem. Go ahead.
Thank you very much for the invitation, of course, to join this webinar seminars for the ISHR. As has been said at the introduction, I'm going to now switch gear and talk about the proteomics landscape of dilated cardiomyopathy. I think I already had a very nice introduction, but just to remind you, I'm Javier Barallobre, and I worked at KCL for the last 14 years, and just very recently I moved to Imperial, that is just across the road. This happened in May, and I also transferred my VHF fellowship with that. And I guess I had the best possible introduction about the extracellular matrix with the talk of Arantxa just before mine, but just to remind you that many diseases, and in fact most diseases, are probably extracellular matrix diseases. And this is not just something that I say, Arantxa says, but also Professor Sennaworth used to say, reinforcing the concept of that the extracellular matrix is important. And for many years I have been using this in the cardiac context and by using proteomics. But as you know, one of the biggest challenges that we face managing heart failure is that patients are usually very heterogeneous. And this is not only concerning the causes, but also the way they are medicated, the associated comorbidities with these patients, and their genetic background. So to study this variability at the tissue level, we established several years ago a collaboration with the Semmelweis University in Budapest, Hungary, and they provided nearly 200 cardiac tissue samples in the numbers that you can see now in the slide. And what we did with those samples was to perform a proteomics analysis of not only intracellular, but also extracellular matrix fractions that we obtained from the tissues. To give you an idea of the scope of this analysis, 200 is actually the total number of heart transplants for any reason that are carried out in the UK each year. We published our results on ischemic patients early, so today I would like to focus more on the dilated cardiomyopathy patients. But of course, to understand DCM, it is also critical to embrace patient heterogeneity. And that's what we started to do by investigating the presence of pathogenic variants first, by performing whole exome sequencing for the cohort of dilated cardiomyopathy patients. With this analysis, we were able to detect the presence of 22 patients with titin truncations, as well as mutations in other genes. And I will concentrate today on titin, laminin alpha-2, and lamin A, as they are the most abundant, and you have them here highlighted in the depiction. What was more surprising for us, actually, was that the presence of these variants, other than being just a complement to the cause of DCM, result in specific compositional signatures. So, for example, if we focus first on sarcomeric proteins and compare for the presence and absence of these titin variants, you can see that only those variants that affect titin, but not the variants in other genes, are associated with changes in the sarcomere. But very importantly, these mutations affect protein levels, but very rarely mRNA expression levels. Instead, if you focus on laminins, which are basement membrane proteins that surround the cardiomyocytes, we could show that only carriers for laminin alpha-2 mutations have disrupted the levels of other laminins, further confirming that the targeted effect of variants on tissue composition, including those that affect the extracellular matrix. Therefore, we can say that specific proteomic signatures can be detected in DCM patients with pathogenic gene variants. However, I would like to stress a very important point, because this is what I've just told you now is only part of the story, because pathogenic variants interact with other clinical factors. The bars that I show you here in red indicate the total number of proteins that were significant within DCM patients when different comorbidities were accounted for. You can see that, for example, while diabetes does not seem to affect much the proteomics profile of these hearts, atrial fibrillation and, in particular, hypertension are key drivers of myocardial composition. But we know, and we've checked in our data, that patients carrying pathogenic variants in lamin A, which is a nuclear protein, have a higher AF prevalence. And therefore, the effect of AF could match the actual contribution of the genetic variants. So that's why it is important to correct for these comorbidities. Thus, proteomics studies are important, they give important information, but they require large numbers, and they require for a consideration of the interaction of pathogenic variants and comorbidities to avoid misinterpretations of the effect of each of them individually. So the next thing we did with our human cohort of heart failure patients was to compare DCM patients to ischemic patients that we had published in a previous paper. And what you can see here in the vertical axis is the ratio of abundance between ischemic heart failure and controls for each protein we detected. And in the x-axis, in horizontal, you can see the same ratio for DCM patients. And from this chart, you can learn the following. That is, ANF is, and not BNP, is the highest and biggest change detected in the ventricle of both ischemic and DCM patients. However, it's very important that we note that ANF levels are very heterogeneous in the dilated cardiomyopathy. And in this chart, what you can see is the coefficient of variance for each protein we identify in our proteomics analysis. And you can see that the variance for ANF is larger than that for any other protein in DCM hearts, including critical proteins that are associated with heart failure. And in fact, we can see that patients in our cohort of DCM distribute in 4 different clusters according to their proteomics profile. And the 2 bottom clusters are mainly characterized by an increase in extracellular matrix proteins and proteins that regulate extracellular matrix assembly. And this cluster distribution also affects ANF levels, and the extracellular matrix protein resembling this distribution of ANF that more closely relates to ANF is versican, which is a large proteoglycan. So what I've been saying to you now is that in cardiac tissues, we can observe that the patients from the fibrotic and non-fibrotic clusters can be separated based on natriuretic peptide levels, but at cardiac level, ANF is more readily detectable than BNP. And this is very interesting as it relates to a previous publication from Upasana Tayal, also from Imperial, where fibrotic and non-fibrotic clusters were identified in these DCM patients using a combination of clinical data and biomarker assessments in plasma, including BNP, as it is the one that predominates in the plasma. Again, and to show this in a more visual manner, DCM patients with high and low levels of ANF are fundamentally different in the levels of this proteoglycan, versican, but they do not differ in collagen content. Failing hearts are also known from previous publications to re-express fetal genes in the ventricle, and the re-expression of ANF is a well-documented example. But the expression of versican also matches these patterns with the re-emergence of genes that are abundantly expressed in the fetal heart. Of course, we know that fibroblasts are the main producers of ECM in the heart, and with the help of Miqueo and Oseda, we explore their single nuclei RNA sequencing data to demonstrate that receptors for ANF are expressed in different fibroblast populations, making them, in principle, responsive to local increases in natriuretic peptides. But this is not the only part of the story. For its degradation and the removal from the heart, versican, this large proteoglycan I've been talking to you about, requires ARANTS5, which is an enzyme that cleaves primarily at the site I indicate here in red. And using CRISPR-Cas9, I generated mice with a mutation at that site, which hinders versican degradation, and of course, because of that, it induces its accumulation. And you can see this in an in vitro experiment. But what was surprising is that these mice show also increased levels of ANF at baseline. And if we infuse angiotensin II for 2 weeks to further increase versican accumulation, this difference is exacerbated with no effect on VMP, suggesting that in addition to stretch, proteoglycan accumulation in the extracellular matrix is an important contributor to the cardiac response to stress, including ANF re-expression and the maintenance of a cardiac fetal reprogramming in heart failure. So to conclude, and as a second summary for my presentation, heart failure is associated with the re-expression of fetal genes. Fetal hearts have highest levels of versican, and the accumulation of this proteoglycan only is sufficient to induce ANF and the ANF re-expression in non-failing hearts without affecting BNP levels, which is the most commonly used cardiac failure marker. And with this, I would like to come to an end, and of course acknowledge current and previous collaborators who contributed with invaluable insight to the work I presented today. Thank you very much.
Thank you, Javier, for your excellent talk. Thank you very much. I would just like to remind the people that the box for question and answer is open, and we will ask questions afterwards. Hello, everyone. It is my pleasure to introduce our next speaker for this webinar series. It's Professor Sandrine Horman. Sandrine is currently professor at the Faculty of Pharmacy and Biomedical Sciences at UC Louvain in Brussels, where she was also recently appointed head of the cardiovascular research pole at the Institute of Experimental and Clinical Research. Her laboratory focused on uncovering the molecular mechanisms underlying cardiovascular inflammation, fibrosis, and thrombosis, with a particular emphasis on non-myocyte cells, especially platelets, endothelial cells, and of course, cardiac fibroblasts. So today she will present some of her most recent and unpublished work with her talk entitled Platelets at the Crossroads of Inflammation and Fibrosis After Cardiac Injury. So please, Sandrine, the floor is yours. You're mute, Sandrine.
Okay. It's okay like that?
Yes.
Okay. So I start again. Sorry for this. So thank you, Alice, for this kind introduction, and thank you to all the organizing committee for this invitation. So indeed, today I will talk you about platelets and how they can influence fibrosis and inflammation in the disease heart. So all of you know platelets for their essential role in hemostasis and pathological thrombosis. For example, when a plaque ruptures, platelets can bind to the injured endothelium and form a clot. And if this clot occurred in a coronary artery, it can block the flow and causes myocardial infarctions. But what few people know is that endothelial injury is not absolutely required for platelet activation. Indeed, platelets can bind to an intact endothelium, and this particularly in the context of inflammation or endothelial dysfunctions. And in that case, the clot is formed by platelets and immune cells, and we talked about immunothrombosis. The interactions between platelets and endothelial cells and leukocytes is possible thanks to specific adhesion molecules and receptors. But platelets have also the capacity to secrete granules, and these granules contain hundreds of different cytokines, growth factors, adhesion molecules, and coagulation factors. And so by this way, they can indirectly influence many, many different processes, including inflammation and fibrosis. Inside these granules, one key molecule is the TGF-beta. Actually, platelets are a major source of TGF-beta, and they can produce about 100 times more TGF-beta than any other cell type. This TGF-beta is synthesized in a latent form with the major active cytokine here in red associated to its latent peptide. And in most cases, this latent complex is secreted outside of the cells where it can be activated by cleavage, for example, by proteases. But platelets and also T regulatory lymphocytes possess a unique mechanism. They express the GAP protein. And this GAP protein can anchor the latent complex at platelet surface. And there, its interactions with specific integrins can induce the mechanical tension that can release the active form of this TGF-beta. And when it is released, TGF-beta can exert its pleiotropic action. It can reduce endothelial activations. It can also reduce inflammations by decreasing the secretion of pro-inflammatory cytokines. And more importantly, this TGF-beta is a really well-known fibrogenic factor. It can promote fibroblast activation, myodifferentiation, increase the synthesis of extracellular matrix proteins. So it's an important mediator of fibrosis and notably of cardiac fibrosis. And because of that, a lot of studies, experimental clinical studies, have tested strategies that block the global productions of TGF-beta. But in most of these studies, there were important toxic off-target effects. In addition, because platelets are an important source of TGF-beta, antiplatelet drugs have also been tested in antifibrotic strategies. And this was done in different experimental models, and the results were quite positive. But here again, there is a limitation with the antiplatelet drugs, which is that there is an increased risk of bleeding. So we have thought that maybe targeting the platelet GAR protein could be a more fine-tuned strategy to block a part of the pool of the TGF-beta and avoid some of the toxic adverse effects linked to its a complete inhibition. And to test this hypothesis, we choose to evaluate the role of this platelet GAR protein in the context of myocardial infarction, most particularly in the post-myocardial infarction response, because it is a condition which is associated with a strong fibrosis and a strong inflammation. So very briefly, 3 important steps: inflammatory, proliferative, and maturation. And directly after myocardial infarctions, there is a strong inflammatory response characterized by endothelial activation, human cell infiltrations, and productions of a high amount of pro-inflammatory cytokines. And once the infarcted zone is clean, this is the resolution step of these inflammations, and then come the repair phase, where the anti-inflammatory macrophages can activate the fibroblast, and these fibroblasts, myofibroblasts, can synthesize the cicatricial fibrosis. And as you know, this fibrotic scare is essential to preserve the integrity of the heart, but scar composition and scar size is also associated with interstitial fibrosis, which will develop in the remote myocardium, cardiomyocyte hypertrophy, and left ventricular dilation, to an adverse remodeling. And this can evolve towards LV dysfunction and in the long term to heart failure. And most of the times, platelets are forgotten from these pictures. However, it is well recognized that platelets can also infiltrate the infarcted myocardium as early as 6 hours after the surgery, and they remain on site until at least 3 days post-MI. But nevertheless, the role of these platelet infiltrations in the post-MI response is not very clear. So to answer these questions, we have generated mice presenting a specific deletion of GARP in the megakaryocyte platelets. We have validated that the production of active TGF-beta by these mice were significantly decreased while the total levels are preserved. And we have investigated the different steps of the post-MI responses from the organ to the tissue, cellular, and the molecular levels. And what you can see is that indeed platelets can infiltrate the infarcted myocardium, and this infiltration is even more important in the absence of platelet GARP, in the PKO mice, platelet GARP-KO mice. But this more important infiltration is not associated with a difference in the infarct size that we have measured by echocardiography or by measuring the plasma troponin levels at 1 day after the surgery. So infarct size is the same, but mortality is different, as you can see. Mortality is more important in the PKO mice. And this is particularly marked in the first week following myocardial infarction, which is typical for infarct ruptures. And indeed, this infarct rupture has been validated by an autopsy of the dead mice. And in the surviving mice, these surviving mice are also different from the control mice, as they show a more important dilatation of the ventricles 14 days after infarction, as it is shown here by the increased end diastolic volume and systolic volume 14 days after infections. And this is associated with a slight decrease in the ejection fraction. So it seems that the hearts from these KO mice are more fragile, and we have tried to better understand the mechanism behind this. And to do this, we have investigated the first step of the post-MI response, which is inflammations. We have performed a RNA-seq analysis at 1 day post-MI on the wild type and the PKO hearts. And what you can see is there is an important enrichment in pathway related to inflammation. More inflammatory response, more production of cytokines, more leukocyte migrations. And the genes there were the most modulated in this RNA-seq analysis were validated by RT-qPCR measurement. And indeed, what you can see is that there is a significant increase in the PKO hearts of IL-1 beta, IL-6, so more pro-inflammatory cytokines, increased expression of E and P selectin, which are adhesion molecules present at the surface of endothelial cells, and, for example, increased expression of MMP8, which is a metalloprotease mainly expressed by the neutrophils. This seems to reflect that there is more endothelial cell activation and more infiltration of immune cells in the PKO hearts. And indeed, these immunostainings show that there is more infiltrated neutrophils and macrophages in the infarcted hearts of these KO mice. And as expected, this exacerbated inflammation is associated with a significant decrease in the expression levels of anti-inflammatory cytokines like IL-10 and TGF-beta. And given that these two anti-inflammatory cytokines plays a very important role in myofibroblast activations, we have, of course, investigated fibroblast properties in these infarcted mice. And as you can show, you can see there is less proliferative fibroblast. There is less myofibroblasts, which are detected by alpha smooth muscle actin staining. And of course, this alteration of the fibroblast properties is associated to an alteration of the scar. A massive drastic decrease in the expressions levels of collagen 1 and 3 with an alteration of the collagen scar, and this is still more obvious with the glycosaminoglycan depositions, which is also an important component of the cicatricial fibrosis. So just to recapitulate, We demonstrate here that the platelet GARP TGF-beta 1 axis is really important in the post-MI response. Its invalidations cause an exacerbation of the inflammatory response with a defect or a delay in the resolution of inflammations, which is associated with an alteration of fibroblast properties. This can cause infarct ruptures or, in the survivors, a more adverse remodeling. And therefore, we believe that the activation of the GARP TGF-beta 1 complexes could be considered as a novel or combined therapy to modulate the inflammatory response and improve cardiac remodeling and function after MI. So with this, I thank all my team and also my collaborators, and of course you for your attention. Thank you very much.
Thank you very much, Sandrine, for that interesting talk about the role of platelets in cardiac fibrosis. And I think Alice and Jonathan, you can probably help me look into the chat box for the questions that have come in. But if I just can start with a quick question for you, Sandrine. I was wondering, do you think that when you do something with GARP, is that all the effects are going through TGF-beta, you think? Or does GARP has independent effects on inflammation or?
No, so this GARP protein is a very specific mechanism of activation of TGF-beta. Now it's not only present at platelet surface, but also at the surface of T regulatory lymphocytes. So by targeting TARP, for example, with antibodies, you are obviously not sure to specifically target the platelets. But in our experiments, we have shown that these T regulatory lymphocytes are involved later in the post-MI responses. So platelets are on the site very early.
Thank you.
We can go check the question and answer box. There's a first question from Gabriele Scattarella for Arantxa. Great talk, Arantxa. Can you elaborate more on the cellular source of endotrophin? Thanks, Gabriele.
So yeah, thanks a lot for the question, Gabriele. So, well, endotrophin is, I wouldn't say ubiquitous, but it's produced by most mesenchymal cells and it's produced by fibroblasts. And interestingly, one of the major sources of endotrophin is the white adipose tissue. So it's very involved in the link between obesity, inflammation, and fibrosis, I would say. Yeah.
Okay, I keep going with the question and answer. It's okay. So there's another question for you from Sudeshna Fish. So being a biomarker lead, I can certainly agree with the point you made about why circulating biomarkers often do not make into clinic. We should discuss this offline, other reasons of offline. But do we have data suggesting that PA1NP is mechanistically linked to action of MRA? Or would you call it more disease biomarker, not specific mechanistically, but suggestive of the disease state?
Okay. So yes, thank you for the question. First of all, there are several issues why biomarkers are struggling in the cardiovascular field, but that would make it almost for another webinar. But of course, happy to discuss offline as much as you want, and that would be great. And regarding the value of P1CP, I think that as it is so downstream the pathway, like in the final steps of the maturation of collagen, I believe that mechanistically MRAs act much more upstream in the inhibition of fibroblast activation and on the synthesis of molecule, and not directly on the mechanism leading to the release of P1CP. So I think that what's reflecting is an upstream activation of the fibroblast and increased production of procollagen. I don't think we can say it's a direct effect on the release of the propeptides.
Okay, so we have more question for Javier. Great talk, thanks. Would you be able to speculate what is the effect of ANF on cardiac fibroblasts? Sorry, I lost it. You mentioned that they have a receptor for ANF.
Yes, thank you very much for the question. Yeah, so as you've seen, I focus more on the opposite communication channel, that is how versican actually makes cardiomyocytes to produce more ANF. It is true that the fibroblasts have the receptors, but what we've been able to do so far is to confirm that although they have the 3 receptors for natriuretic peptides, the one that is more specific for natriuretic peptide A is the least abundant in cardiac fibroblasts. So it's difficult at the moment for us to speculate what would be the effect, because anything we do in fibroblasts directly might actually be overloaded with amounts of ANF we can put. So there are different receptors that actually can have suboptimal binding to ANF. So what I would say at the moment is that we know what happens when you have too much versican. If you have too much ANF on fibroblasts, we can speculate, and most people have said that this is antifibrotic, but whether this is a direct effect or a second round effect is more difficult to confirm at the moment.
All right, thanks.
And I'll just follow up because I have one question for Javier. So in the dilated cardiomyopathy, the patients clustered into 4 different proteomic clusters, and they also had variability in ANF levels. Do you think that VeriScan and the processing of VeriScan by Adam TS5, that different cleavage factors of VeriScan are actually leading to different cellular signaling that are leading to those 4 different proteomic clusters?
Thank you. So these proteomics clusters, first of all, I need to disclose that this is not based only on extracellular matrix. So we did 2 layers of analysis. The first layer was intracellular, and that's what we used to generate our 4 clusters. And then from those clusters, we saw that some of them have a more profibrotic profile, and the others have a least profibrotic profile. Yes, there are enzymes, I've studied them before, like arantiases, that can be the cause of, or at least it can regulate the levels of proteoglycans. But it's, of course, difficult to say whether the levels of versican will be just a direct consequence of more or less enzyme, because these enzymes are not only produced in the heart, they are produced everywhere. So it would be difficult to say that this is only due to the presence of enzymes. They contribute at a local level, they are critical, but whether they are the only contributing factors, I think it would be too much saying.
Okay, thank you very much, Javier. So we'll go back to the question and answer box. So our next question is to the first. Well, let's go to the third speaker just so the third speaker can have some questions. So the question's from Raja Amir Thanlingaman. And the question is to Sandrine. So you showed how GARP latent TGF beta 1 complexes might be targeted to improve post-MI remodeling. How do you think this approach can balance the beneficial repair effects of TGF-1 beta with its potential to drive fibrosis and long-term left ventricular dysfunction?
Yes, thank you. It's a very important question, but I think that the timing in the post-MI response is something really important. So here our hypothesis is that this platelet TGF-beta activated via GARP is important to control endothelial activations and inflammatory response. And it's really crucial to limit this inflammation. It's important for the post-MI remodeling. Of course, TGF-beta is also produced later, probably not by platelets because they are present very early. And this TGF-beta, which is involved in fibrosis, and again, cicatricial fibrosis is really important. It's a good thing to preserve the integrity of the heart, but later you have the development of interstitial fibrosis, and TGF-beta is also involved in that. And clearly, of course, it's important to counteract the action of this TGF-beta. But I think that in the very early response, TGF-beta produced by platelets plays an important role to limit the inflammatory response.
Okay, thank you very much. And we have one more question for Sandrine from Magu Anje. So what is the impact of hypertension on GARP and TGF-beta release from the platelets?
So thank you. Also, it's something we are investigating, the role of this GAP TGF-beta axis, but in other pathological conditions. So regarding hypertension, I have not the answer yet, but what I can say is that in models of pressure overload, platelet GARP is also involved. There is also an infiltration of platelets in the myocardium, and we are currently investigating the impact it can have maybe on the development of interstitial fibrosis in this condition.
We also have one last question for Arantxa. And it's, is there variability in the PICP levels due to extra cardiac fibrosis, for instance, liver fibrosis or lung fibrosis? And how might that affect its specificity for cardiac remodeling?
Yes, thank you very much for the question, which in this is a critical point. And this is one of the limitations of the biomarkers of fibrosis that we have, is that, as also Javier was mentioning, fibrosis is, major contributor of disease all over the body, and we don't have specific biomarkers of cardiac fibrosis. And indeed, this is something that we have to take into account when analyzing circulating biomarkers of fibrosis, that P1CP can be influenced. Not so much, studies don't show it a huge impact of lung fibrosis, but yes, by liver fibrosis and also by bone metabolic diseases, which are also highly collagen type 1 dependent. So the comorbidities of the patients are also something that we should take into account when analyzing the value of these biomarkers, yes.
Okay, I think that we're good also for the timing, and I would like to thank all our speakers for their excellent presentations, and of course also the participants who join us online. I would just remind you that this was a part of a seminar webinars organized by the ISHR, ECI, and MCI, and we look forward to bringing you more in the coming months. So please stay connected through our social media channels for more ISHR information. And thank you very much again for the participation. And thank you all.
Thank you. Bye-bye.
Bye-bye.
Bye.
Thank you. Bye-bye.